Let's take a look at review for Part D of the Regents, which is the lab component, and this is diffusion and osmosis. If you remember, we made a model cell using dialysis tubing, and we filled that cell with a starch solution and a glucose solution. Okay, this would be the diagram that you filled out. And then what we did was put iodine in the beaker of water so that we could use it as an indicator for starch. Remember, starch is a complex molecule, so it's very large. If we look down here at the two cells, we can see that this is the beginning cell, and then it turned into this cell, which is a very dark color. Remember that iodine in the presence of starch, okay, tells us it's present in positive tests when it turns a blue-black color, or the color that you see here. That tells us that there is starch present inside the cell. Well, where did that iodine come from? It came from the water in the beaker. That's where the iodine is. So it was able to move through the membrane into the contents of the cell. Next, remember we took a dropper and took some of the solution out of the beaker and put it into a test tube. And then we put some Benedict solution in it. Remember, Benedict is a blue color to start with, and it's an indicator for sugars. Then we put it in a hot water bath and observed a color change. Well, there was a color change because it was a positive test for glucose. The glucose was small enough to diffuse from the inside of the cell to the outside in the solution in the beaker. In order to observe osmosis, we used red onion, we made a wet mouth slide, and then we applied salt water to the slide. And to put salt water on the slide, we took the dropper and put it on the edge of the cover slip and then put a piece of paper towel on the opposite side so that would draw it underneath the cover slip. So remember that we're talking about a solution that is going to be higher in solute outside the cell. So we have the water movement is going to go from inside the cell outside the cell. So the cells appear to shrivel or shrink. Okay, so that would be a hypertonic solution.